I want to thank the organizers of PCE3 for inviting me to give an overview of Session 4 on prebiotic complexity. There's a number of topics that I think are current in today's research. I'm just going to give a little list of these because the speakers later on will be touching on these as well. I think we need to get a little bit more information about possible sources of organic compounds on the early Earth. We have to decide which could be synthesized, as Stanley Miller suggested in 1953, and which could be delivered, and whether it even matters, because no matter how they come to the early Earth, they would join a mixture of materials and start to be altered chemically and physically. We need to talk about sources of energy. Light, redox reactions, wet-dry cycles, all of these are possible sources of energy that would drive some of the processes required for the origin of life. Then there's the role of water. Condensation reactions produce polymers. Hydrolysis degrades polymers back to their subunits. There would have been a balance between condensation and hydrolysis, so we need to understand both of those processes. Self-assembly is very important in life today. Self-assembly of membranes, self-assembly of proteins during folding, self-assembly of duplex structures of nucleic acids. These are all part of life today and would certainly be part of the origin of life because the self-assembly is a physical rather than a chemical process and just happens spontaneously. We need to understand how polymers could be synthesized because they would turn into the structural and catalytic polymers of early life. We really don't know too much about how polymer synthesis could occur, and there's a lot more to learn. Compartments. Were they organic compartments to keep systems of molecules together? Or perhaps were they mineral compartments, such as been proposed for hydrothermal vents? Then we need to understand the assembly of systems from simple to complex. This is for the future, I think. We don't know how that could have happened yet, but I think it's a very important topic to begin to think about. Because as you assemble systems, we will get up to the point where primitive metabolism and primitive genetic information could emerge to give rise to life as we know it. Finally, we need to understand the first steps of evolution in populations of protocells. That is certainly for the future. It hasn't been worked out yet, but I really see that as a future goal of research in this area. So life began by assembly of certain organic molecules, and we can learn from how organic molecules self-assemble into living structures today. Certainly nucleotides are the monomers of nucleic acids, and nucleic acids are the carrier of genetic information in life today. Amino acids polymerize into proteins, and of course proteins are prominent in all life today, both as structural and catalytic properties. Finally, amphiphilic compounds self-assemble into membranous compartments. That must have gone on the early Earth to give rise to cellular life. So where did organics come from? Jim Cleaves is going to talk about that. I think a clue to that question is that all the biogenic elements arrived during accretion of our planet. Some of these as organic compounds. So when we think about sources of organic compounds, we have to decide between those that could have been synthesized in the early Earth environment. Stanley Miller's paper in 1953 really sparked this understanding, but George Cody and Palmer and Sutherland have also advanced the field about synthetic organic compounds. Then another set of compounds could have been delivered, and Keith Van Folden in 1971 analyzed the Murchison meteorite. And I'll touch on that because that gives us a clue to what kinds of compounds would have been delivered that are relevant to the origin of life. Chaiban Sagan had a wonderful paper in 1993 showing that, in fact, interplanetary dust particles were a major component of organic compounds being delivered to the early Earth. And I think we have to see, um, how much delivery was part of the process by which compounds were made available. So let's just quickly summarize what we know about the origin of the solar system and of the Earth. 4 billion years ago, the sun, the early sun, was in a molecular cloud and was beginning to glow. Planets were beginning to form from a cloud of dust and gas. And we can just think of that as stardust, in fact, because these are the ashes of stars that have gone through their life cycle. And the important thing about those dust particles, and one is shown here in the center of the slide, is that there would be an ice layer on the surface of the particle, and that ice layer would have in it water, methanol, carbon monoxide, and ammonia. We know that from radio astronomy and infrared astronomy. Lou Alamandola and others have shown that ultraviolet radiation can produce a photochemical reaction leading to more complicated molecules. Those molecules include things like amino acids, purines, hydrocarbons, and amphiphilic molecules. Those then would have come along during accretion of the dust into comets and planetesimals, with a lot of interplanetary dust particles still circulating in our solar system, and those would then have been delivered to the early Earth during late accretion after the ocean had formed on the planetary surface. So this is what a dust particle looks like. This is work from one of the ESA laboratories, and it shows a interplanetary dust particle with the green circles showing where organic compounds were detected on this dust particle. So I think we can be pretty certain that, in fact, interplanetary dust particles did bring in a lot of organic material. Brownlee has suggested that even today, something like 30,000 tons of silicaceous dust particles come into the atmosphere every year, bringing along with it some of the organic compounds, just as would have happened at many orders of magnitude faster on the early Earth. The way we understand what these compounds were is by studying carbonaceous meteorites. And there's a little town in southern Australia called Murchison. And in September of 1969, the residents of Murchison were startled when a bolide, a fireball, appeared in the sky and exploded, dropping 100 kilograms of fragments of the original meteorite all around the fields of Murchison. Those were picked up by townspeople and very quickly by scientists as well. Here's one fragment that came through my lab. It's about the size of a golf ball. When we break open a sample like this, it turns out that we can analyze for organic compounds. And here's a sort of a simplified version of the kinds of organics that are in the Murchison meteorite. So there's about 1.5% of insoluble polymer, organic polymer, called kerogen. More interesting are all the soluble organics, and those are the ones that really have something to do with life processes. About half of those soluble organics are carboxylic acids, starting with acetic acid and going all the way up to monocarboxylic acids, 12 and even 13 carbons in length. These are soap-like molecules, by the way. Dicarboxylic acids are present. Amino acids are there, about 50 parts per million of maybe 70 or so different amino acids, 10 of which, in fact, are part of proteins today. There were amides present, aliphatic and aromatic hydrocarbons, aldehydes, alcohols, amines, and even some purines, about 1 part per million of purines like adenine and guanine. So these compounds I think of as the molecular parts list required for life to begin. So let's just come up with some conclusions about this part of my overview. I think we can be pretty confident that organic compounds were in fact delivered to the early Earth. Synthesis has been demonstrated in laboratory simulations, but there's no direct evidence that organic compounds were also synthesized on the early Earth, although it's a pretty good assumption, I think. It may not even matter because any organic compounds delivered or synthesized would immediately begin to altered and mixed by chemical and physical processes. What energy sources? Well, light energy is by far the most abundant energy today and drives photosynthesis and virtually all of life on Earth. But what pigments and photosystems might have been available to transduce light energy into chemical energy on the early Earth? This is an open question. We really need much more work on how to capture light energy using plausible pigment systems that would have been available on the early Earth. Chemical energy was, of course, available. We know that from our studies of hydrothermal vents, where there's obvious sources of chemical energy, and that's why they're so attractive as a place to consider whether they were a site for light to begin. But again, the chemical energy had to somehow be coupled to biological processes to develop into a primitive metabolism. Finally, evaporation on land masses can concentrate potential reactants, and as the evaporation occurs, water activity is reduced, and this can drive spontaneous condensation reactions. And I've often thought that this energy could drive polymerization of monomers like amino acids, even nucleotides, into primitive polymers. Liquid water is essential. About 99% of all water today is in the ocean, and it just makes it pretty obvious that life had to begin in salty seawater. But there is an alternative water which is distilled by evaporation from salty seawater and precipitating on volcanic land masses. I think it's plausible that life could have begun in fresh water, and there's some pretty good evidence suggesting that that's a distinct possibility. When we think about hydrothermal vents, that's going to be what Laurie Barge is talking about in this session. It was proposed by Corliss and Bross and Hoffman way back in the early 1980s that life could have begun there, extended by Mike Russell and Bill Martin in the 1990s, and more current laboratory simulations have been carried out by Barry Hershey and Nick Lane's lab and Burkhar in 2015, and of course Laurie Barge, very active in this right now. Of course, alkaline hydrothermal vents are produced by serpentinization, and we can imagine that something like that might be happening on the moons such as Enceladus, the icy moons of Enceladus and Europa, leading to a possibility that life might be present on these icy moons. Now here's a speculation by Lane and Martin about how a pore that has a kind of a mineral membrane might have been involved as a source of chemical energy leading to a primitive metabolism. On the left, you see a very thin mineral membrane composed of metal sulfides, and you also know that hydrogen was available in the vent fluids, and this hydrogen could have reduced in a catalyzed reaction carbon dioxide into things like the compounds shown in that little line across the bottom, finally leading up to molecules that could have been very much involved in a primitive metabolism. Keep in mind there's also a pH gradient present. The alkaline vent fluids are very alkaline, pH 9 and above, and yet the early ocean was probably fairly acidic, like pH 6. That allows a chemiosmotic source of energy, which is really very interesting possibility. Lane and Martin imagined that as passage of time, a lipid bilayer might have accumulated on the mineral membrane, producing a permeability barrier to protons, and that a simple version of an ATP synthase might have actually been driven by this chemiosmotic energy source. So I think that's interesting ideas. They need to be followed up, and it's a very nice place to have research go on. An alternative salty water is one that we've been working on, that is freshwater hydrothermal fields. An example is our field site called Bumpus Hill in Mount Lassen, just a few hours drive from where I live in Santa Cruz. And we've often been up there. We're actually trying to do experiments in these sort of freshwater environments. Mukajanian was one of the first to propose the origin of cellular life, the terrestrial anoxic geothermal fields, PNS paper in 2012. And Bruce Deamer and I just came out with a hot spring hypothesis published in a recent issue of Astrobiology. If you're interested, you might want to look into that. Well, the nice thing about freshwater hot springs is that there are fluctuating interfaces. This is in contrast to the one mineral water interface of hydrothermal vents. There's three fluctuating interfaces: mineral water, mineral atmosphere, and atmosphere water. There's also abundant light energy. This would have allowed an early development of photosynthesis if pigment systems were available in these environments. Finally, we've pointed out that wet-dry cycling is in fact a source of chemical energy that can drive polymerization. So this is a relatively easy place to work because these kinds of sites are available for research all over the world. I've been to Kamchatka, Russia, working on this sort of a hot spring area. I've been to New Zealand in February of this year. Bruce Damer and I were down there doing experiments. And of course, Hawaii and Iceland all have interesting freshwater hydrothermal fields to do research on. So what about polymer bond synthesis? I hinted that condensation reactions could be driven by wet-dry cycles, and let's just understand why that is. If we mixed a little ethanol and acetic acid, pure compounds, and let it go to equilibrium, we would discover that at equilibrium, they had spontaneously formed an ester bond that links them into ethyl acetate with water as a byproduct. What we normally don't think about is that this could also happen in things like nucleotides. Mononucleotides have an acidic phosphate group, they have an OH group on the ribose, and in principle, there should be dimers formed. Very small amounts, about a 1,000:1 ratio of monomers to dimers. If we waited long enough, it's going to be a very long kinetic process, but it should occur. And if we drive that by removing the water, we can certainly pull the reaction toward polymer formation. The same thing can happen with amino acids, and Lee Cronin has done a lot of work on this. Two glycines can come together to form a peptide bond, and if we can pull away the water, we can pull that reaction to the right and make peptides, maybe up to 10 or so amino acids in length. So how do we get rid of the water? We simply let it evaporate. When water leaves a solution that is undergoing a condensation reaction, the reaction is pulled to the right and you get increasingly long polymers formed. So I think that's a very useful way to think about how polymers could have been synthesized spontaneously on the early Earth. So Luke Lehman and Christine Keating are the last two speakers in this session. And the title of their area is Processes Acting on Building Blocks, Assembly and Complexification. So self-assembly processes include membrane formation, protein folding, and duplex nucleic acid structures. These all should be part of what we bring to bear as principal driving forces of self-assembly that could be occurring on the early Earth. You've all seen this occurring. Anybody that's blown a soap bubble has in fact made a membrane out of fatty acids that are soaps. And we found years ago now that, well, let me say first that membranes are composed of amphiphilic molecules. An amphiphile is just a molecule like a soap that has a long hydrocarbon chain and a hydrophilic head group. And there's a fatty acid shown in the upper organic compound illustrated here. On life today, these have been evolved into more complex molecules called phospholipids and cholesterol And these are biological amphiphiles that self-assemble in all cell membranes today. Years ago, we showed that that you could watch this under the microscope, and the self-assembly is just beautiful to watch. This is a little bit of a time lapse to speed it up about fivefold. And we have dry phospholipid under the microscope on the left, and we've added water on the right. And you can see the water penetrating into the dry lipid, and as the lipid is hydrated, it squeezes out first into tubular structures, and then the tubules break down into membranous vesicles. And you can see that little spider-like structure in the center of the field, and you can actually watch little vesicles popping off of the ends of the metastable tubular structure. So that's really fun to watch, and this all happens spontaneously. All you need is a dry membrane-forming lipid and some water to see this for yourself. We saw this with carbonaceous meteorite extracts from the Murchison meteorite. We found that, in fact, there are soap-like molecules in the meteorite that can self-assemble into microscopic vesicles. This is what they look like. They really are membranes because we can encapsulate fluorescent dyes, as you see on the right. So I think it's a very plausible that such such compartments would have been available for the origin of cellular life. One last thing I want to point out about lipids is that they have a really interesting process by which they can encapsulate really big molecules. This is done by drying them down to form a multilamellar liquid crystal. And all you have to do is to dry down a mixture of lipid vesicles in the presence of solute molecules as big as proteins or nucleic acids. The solubles are captured between layers of the multilamellar matrix that is formed when the vesicles fuse. When you rehydrate the vesicles, you discover that the molecules that were originally outside have now been captured inside. This is what it looks like. On the left is a freeze fracture image of lipid vesicles. It's just a phospholipid. And when you dry it out, you see that multilamellar lipid phase that forms, that captures the molecules. We know for a fact that molecules are captured because in 2013 we published a paper with Laura Topozzini as first author. And Laura did X-ray diffraction showing that AMP forms a paracrystalline array between lipid bilayers in that multilamellar matrix. So that's an organizing principle that has hardly been explored. And I think that can tell us a lot about the polymerization process that can spontaneously go on as we go through wet-dry cycles. Finally, let's end up with an RNA world. We all kind of know what that means, that RNA used something like RNA as a catalyst and also a way to store and replicate genetic information. And it goes all the way back to some of the original workers in the field, Francis Crick, Leslie Orgel, Carl Woese. Wally Gilbert came up with the term RNA world. And we can imagine that maybe RNA was very much involved in the origin of life. So a lot of people have been working on this, and it's a very nice working hypothesis. It's inspired a lot of experimentation. Bartel and Shostak, for instance, in 1993, showed that ribozymes can be isolated, evolved from a large pool of random sequences. To me, that was really a wonderful paper because it answered the question of how information could be embedded in the sequence of bases in RNA as you began to select and evolve, in this case, for a ligase activity. Atwater et al. And Phil Holliger's lab has shown how ribozymes can evolve in a eutectic mixture, including ice. Then Horning and Joyce have amplified RNA by an RNA polymerase enzyme, PNAS article in 2016. These are really nice papers, very exciting, but it begs the question, how can a large pool of RNA polymers be synthesized so that selection and evolution can begin? That's another of the open questions in our field. And I urge everyone listening, if you're interested in this, there's so much that remains to be done trying to address that question. Well, I'm going to end up with a hypothetical scenario that builds on what I just told you. It seems to me there must have been a non-enzymatic process by which potentially catalytic and replicating polymers were produced on the early Earth to initiate this primary process that is now part of all biology, replication and catalysis. There must be a way to encapsulate these polymers in membranous compartments to form vast numbers of protocells. Each protocell is in fact a kind of a microscopic experiment in a natural version of combinatorial chemistry. A few of these would happen to contain combinations of polymers that could have these properties of catalysis and reproduction. I just love it if somebody can make that happen and discover that a protocell is in fact an evolving entity capable of undergoing selection. Finally, the selection caused competition to evolve toward the first cellular life. And I think it's a pretty good possibility that we will be able to see something like this happen in the laboratory, getting pretty close to that. So with that, I will close my overview and go on to the next 4 talks. Thank you for your attention.